Assalamualaikum and good day to everyone. Welcome back. My name is Dr. Wang. The topic that I would like to present to you today is X-linked disorders. Here are the study outlines. At the end of the lecture, you should be able to understand and describe the differences between autosomal disorders and sex-linked disorders. Explain the inheritance patterns and characteristics of sex-linked disorder, which include X-linked dominant, X-linked recessive, and Y-linked. Know the examples of sex-linked disorders and describe X inactivation and bar body. What is X-linked disorder? It is a pattern of inheritance in which the transmission of traits, characteristics, depending on the presence or absence of mutated allele on the X chromosome of the sex chromosome. When one mutated allele is present on the X chromosome in male, the phenotype will be expressed. This is because male only has one X chromosome in the sex chromosome. However, female with one mutated allele on the X chromosome are carriers and generally do not express the disease. X-linked disorders can be categorized into dominant or recessive. Here are the characteristics of X-linked dominant disorder. It is caused by mutation in genes on the X chromosome of the sex chromosome. Therefore, one mutated gene is sufficient to manifest the condition. Affected father will pass the disease 100% to their daughters, but not to their sons. There is no male-to-male transmission. Father cannot pass the disease to their son. Women carrying a mutated allele will express the symptoms of the disease. However, female may express less severe symptoms compared to the male because female has another copy of X chromosome which is not affected. This diagram shows a Punnett square analysis of X-linked dominant disorder for an affected father and an unaffected mother. Since it is a dominant disorder, we assign capital A as the abnormal or mutated allele and a small a as the normal allele. It is an X-linked disorder, therefore, we also need to assign the gender in the Punnett square analysis. XY for male and XX for female. Father is affected, therefore, we assign an abnormal allele on the X chromosome for the father. The mother is unaffected, therefore, the genotype for mother is homozygous with a small a. When affected father married to an unaffected mother, the father will pass the abnormal allele on the X chromosome to his daughters and Y chromosome to his son. Therefore, the probability of their offspring to inherit the disorder is 0% for sons and 100% for daughters. This diagram shows a Punnett square analysis of X-linked dominant disorder for an unaffected father and an affected mother. Since it is a dominant disorder, we assign capital A as the abnormal or mutated allele and a small a as the normal allele. It is a X-linked disorder. Therefore, we also need to assign the gender in the Punnett square. XY for male and XX for female. Father is unaffected. We assign a normal allele for the father. Mother is affected. We assign an abnormal allele for the mother. The genotype for mother is 1 capital A and 1 small a. The genotype for mother is heterozygous. This is because it is very rare for a person to have both abnormal dominant alleles, as people with two dominant abnormal alleles usually won't be able to survive. When an unaffected father married to an affected mother, the mother will pass the abnormal allele either to her son or daughter. Therefore, the probability of their offspring to inherit the disorder is 50% for both the sons and the daughters. Here are some of the examples of X-linked dominant disorders, such as Fragile X syndrome, incontinentia pigmenti, congenital generalized hypertrichosis. In the next few slides, I will present some examples of X-linked dominant disorders. Fragile X syndrome is one of the most common causes of mental retardation in male. It is caused by mutation in the Fragile X mental retardation gene, or FMR. FMR protein is essential for normal neurodevelopment. The mutated gene will cause the expansion of the CGG repeats. Affected individual with the expanded CGG repeat will have reduced transcriptional expression of FMR gene, therefore lead to reduced or blocked production of FMR protein. The characteristics of Fragile X syndrome are such as elongated face, broad forehead, large prominent ear, strabismus, which is crossed eyes, highly arched palate, hyperextensible joint, and so forth. Incontinentia pigmenti is caused by mutation in the inhibitor of kappa-like polypeptide gene enhancer in B cells kinase gamma, or IKBKG gene, which regulates a family of transcriptional factors. Affected females have abnormal development of skin, hair, teeth, nails, brain, and eyes. The disease is fatal in male, but not in female. This is because male only has one X chromosome. An affected newborn girl will have pus filled vesicles on her arms and legs. The lesions will become warty and then turn into permanent brown splotches. Affected individuals also experience hair loss, scarring of the retina, seizures, and underdeveloped teeth. Congenital generalized hypertrichosis is an X-linked dominant disorder. People with this disorder have an abnormal amount of hair growth on the body. Male exhibits excessive facial and upper body hair. Female exhibit less severe asymmetrical hair distribution. However, the palm, soles, and mucous membranes are not affected. I have described 3 examples of X-linked disorders here. You are not required to memorize the name of the mutated genes and the characteristics of these disorders. This is just for your knowledge. What you need to remember is the examples. I have explained the characteristics and examples of X-linked dominant disorders. Next, I would like to explain the characteristics of X-linked recessive disorder. For X-linked recessive disorder, one mutated copy of a gene on the X chromosome is sufficient to manifest the condition in male. There is no male-to-male transmission. Father cannot pass the mutated allele to the son. Disorder is primarily present in male. This is because male only has one X chromosome. Affected males pass their mutated alleles to their daughters. Female carriers can express phenotypes at variable levels. This diagram shows a Punnett square analysis of X-linked recessive disorder from an affected father and an unaffected mother. Since it is a recessive disorder, we assign the capital A as the normal allele and a small a as the abnormal or mutated allele. Father is affected. We assign an abnormal allele for the father. Mother is unaffected. We assign the normal allele for the mother. The genotype for mother is homozygous. When an affected father married to an unaffected mother, the father will pass the abnormal allele to his daughter, and mother will pass her normal allele to all her children. Therefore, the probability of their offspring to inherit the disorder is 0% for their son and 100% as carrier for their daughters. This diagram shows a Punnett square analysis of X-linked recessive disorder from an unaffected father and a carrier mother. Since it is a recessive disorder, we assign the capital A as the normal allele and a small a as the mutated or abnormal allele. Father is unaffected. We assign a normal allele for the father. Mother is a carrier. Therefore, the genotype of the mother is heterozygous with one normal and one abnormal allele. When an unaffected father married to a carrier mother, the father will pass the normal allele to his daughter, and mother will pass the abnormal allele to her children. Therefore, the probability of their son to inherit the disorder is 50%, and 50% as carrier for their daughters. The examples of X-linked recessive disorders are such as hemophilia, Duchenne muscular dystrophy, and red-green color blindness. Hemophilia is a classic example for X-linked recessive. It is caused by a mutation in the factor VIII gene, in which the protein produced are needed for blood clotting. People with this disorder has a tendency for bleeding. A hemophilic child could not play sports because if he fell, he could sustain a cut that bled for several weeks. In the olden days, whole blood replacement was the only treatment. Many children with hemophilia die from kidney failure due to the large amount of fluids they received during transfusion. In the recent years, people with hemophilia usually treated with blood products such as factor VIII, which is not found in hemophilics. The pedigree chart here belongs to the European royalty. Queen Elizabeth is the proband. She passed the mutated hemophilia gene to some of her descendants. The next example is Duchenne muscular dystrophy. It is caused by a mutation in the dystrophin gene, which codes for protein dystrophin. Dystrophin protein is essential in stabilizing cytoskeleton during muscular contraction. People with this disorder will not be able to synthesize dystrophin protein. The average life expectancy for this disorder is around 25 years old. Red-green color blindness is another classic example of X-linked recessive disorder. People with this disorder is unable to see color under normal light conditions and have difficulties in differentiating red and green due to the absence of the red-green retinal photoreceptors. Another type of color blindness called blue-yellow color blindness. People with this disorder is unable to differentiate between blue with green and yellow with violets. Blue-yellow color blindness is an autosomal disorder as the mutated gene is located on the chromosome 7 and it is not sex-linked. Y-linked disorder is only affect male. It is caused by mutations on the Y chromosome. Y-linked disorders are rare and it only passes from father to son. Some examples are such as male infertility and hypertrichosis. The next topic I would like to discuss is on gene dosage and the Lion hypothesis. For sex chromosome in human, females have 2 X chromosomes and males have only 1 X and 1 Y chromosome. The X chromosome is about 5 times larger than the Y chromosome. Therefore, if females express all the genes on both X chromosomes, they will express more genes than male. How are humans going to overcome this? In order to keep the gene dosage equal between male and females, a process called X inactivation occurs. X inactivation is a process in which one of the X chromosomes in female is inactivated by coiling and condensing. When condensed, it cannot be transcribed, therefore cannot produce messenger RNA. The condensed X chromosome is known as bar bodies or sex chromatin bodies. The inactivation occurs randomly. It could be the X chromosome from the paternal or the maternal. After X inactivation, only a small piece of the inactivated X chromosome, which is known as bar bodies or pseudo-autosomal region, is transcriptional active. This X inactivation process is hypothesized by Mary Lyon, an English geneticist. Therefore, it is also called the Lyon hypothesis. X inactivation occurs in early embryonic development. The X inactivation process requires a gene on the X chromosome called X inactive specific transcript or XIST. After inactivation, females have only one functioning X chromosome, same as the X chromosome in male. The inactivated X chromosome of female will have the same gene dosage as the male Y chromosome. Once inactivated, the X chromosome will remain inactive throughout subsequent mitotic divisions. Bar bodies are only reactivated during mating to continue the cycle of inheritance. Genes in the pseudo-autosomal regions are present on both sex chromosomes. As a result, men and women each have 2 functional copies of these genes. Many genes in the pseudo-autosomal regions are essential for normal development. The image here shows the bar body of an inactivated X chromosome. Woman has 2 X chromosomes on the sex chromosome. One of the X chromosomes is randomly inactivated and become bar bodies. For trisomy X disorder or 47XXX, there will be 2 bar bodies. This is because trisomy X has 3 X chromosomes. 2 of the X chromosomes will undergo X inactivation and become bar bodies. This is the end of my lecture. Thank you for listening. Let's go through some quiz. Regarding X-linked recessive inheritance. A. Disorder occurs in every generation. False. B. It is a male-to-male transmission disorder. False. C. One mutated allele on the X chromosome is sufficient to express the phenotype in male. True. D. If father affected and mother unaffected, all their daughters will be a carrier. True. E. Thalassemia is an example. False. X-linked disorders A. Are polygenic. False. B. Follow the Mendelian trait. True. C. Are caused by mutation of the gene in autosome. False. D. Are diagnosed by DNA sequencing. True. E. Are primarily present in males. True. A 26-year-old woman who is a carrier of hemophilia is married to an unaffected man. They plan to start a family and went for genetic counseling. The most likely probability for them to have an unaffected child is, based on this diagram, the answer is 50%. In a family, the father is having a genetic disorder, but the mother is unaffected. The disorder is inherited by the daughters only and not the sons. The mode of inheritance for this disorder is most likely C. X-linked dominant.